Okay, good afternoon, everyone, on this EXIMIOUS Symposium. Welcome. I'm Peter Hoed, trying to host this symposium. It's our fourth EXIMIOUS Symposium, which is on chemical exposures via skin. So the title is, as you know, Do Chemicals Penetrate Human Skin? And how skin absorption and chemical contributes to human exposures. So just first, I will have a brief introduction on what is EXIMIOUS and what you may expect from this symposium, and then I will hand over the parole to the speakers of this symposium. So first of all, a few small housekeeping issues. And I need to pay your attention to the fact that we record this event and that it afterwards will be shared via our website, and therefore you can have a review of this seminar later on if you think some issues you want to see back, or a colleague wants to see what has been said this afternoon. And then I also want to pay your attention that this, as you did see, the platform we use is Teams, and so to communicate, we suggest that you or use the chat or the Q&A function. In the chat you can leave a remark, but there we would like you to use the chat mainly for technical issues. If you don't hear us, if there is a problem with the progress of the screen and so on, so then the people behind the screen will try to solve this problem. And the Q&A function is mainly then for really the questions concerning the content of this seminar. So if you have a question to speakers, you can post there your questions, and then we will try to answer to these questions during after the presentation of each speaker, and your question can then be answered by them, or later on, if needed, we can have also a written answer. So just to introduce EXIMIOUS. EXIMIOUS is a Horizon 2020 project on the mapping of exposure-induced immune effects. So connecting or trying to connect the exposome and the immunome. So as one hand, we as humans are exposed externally and internally via different routes, via our diet, lifestyle, environment, our work, and so on. And this will have an effect on our immune system and will generate an internal exposure by antibodies. Different cells will be activated and so on, or tissues, proteins, and this will certainly also depend on our genetic background. And that is what we try to bring together in this project. And so the project is already running some time, but as you know, we also had a COVID crisis, which has made it very difficult to sample in real life. But we trying to cope with this and trying by working hard to get our samples as we anticipated in the beginning of the project. It has also a relatively big budget, as you see, but it is also a project with quite some partners, 15 partners in 7 countries, sampling different cohorts. I will not go in detail of which cohorts we sample, but just to give you a view on the flow of the project is it's really multidisciplinary and a multi-omic approach where we have one hand exposure in real world, and we try to collect not only exposure but also health status of people at different stages in their life, and sample different levels, the genome, epigenome, the transcriptome, and so on, and bringing all this data together in an integrated database, and then using different statistical but also artificial intelligence tools to link the exposures to disease and to come to different tools, biomarkers, disease markers, and hopefully also a better insight in mechanisms how certain exposures, depending on maybe our genetic background and or our genetic background and immune related diseases. And so this slide just to give you a view of who is involved in this. So the different research groups, the 15 groups are placed here, and the different countries in red stain on the map of Europe being involved in this project. But EXIMIOUS is not the only project in Europe trying to do exposome research. So we are in fact 9 different projects, all bundled in the European Human Exposome Network, or in brief EHEN, and all the different projects are given here, and their logos in alphabetic order, just to give you a view on the different projects. And if you're interested, I cannot go in details on what their typical focus is. You can find them. They all have a separate website, or you can find our joint website of the European Human Exposure Network, as you see here below. So today we have our 4th EXIMIOUS Symposium, and we had already therefore 3 other symposia. Just gave you here the titles, and you can find them if you're interested on the content on our website, EXIMIOUS H 2020 EU. So the first one was exposome immunome interactions, as more an introductory symposium. Immune-mediated disorders, so somewhat deeper view on the immune system and immune differentiation linked to age and exposure. So all these first 3 symposia were more linked to the immunome pillar of our project. And today we have somewhat a different theme. So our title, as I said, is "Do Chemicals Penetrate Human Skin?" And how skin absorption can contribute to human exposure. And so why did we set up this symposia? Because we know or think that skin exposure is somewhat a forgotten route of exposure in general, although we all know that skin is important and so on, but it is more difficult to measure than oral uptake and inhalation. And it is not always so clear how much of a chemical particle that we come in contact to will really penetrate the skin. So it's also a quite difficult route of exposure, and therefore we have built this symposium, not too long, so it's 2 hours, on first of all in the first talk on pharmacotoxicokinetics, so looking into the internal exposure and how the exposure to the skin leads to systemic exposure. And then we have the 2 next sessions in this symposium is two examples, in fact, one on metals and the other one on PAHs due to contact via the skin. So as I said, I'm Peter Hoed, giving you this brief introduction, but we have the three other speakers. So the first is on ADME and toxicokinetics, which is pivotal in this exposure, and it's in all exposure routes, but in skin it is certainly a very important issue. It's a bit, and the toxicokinetics is really a missing link between exposure and the toxicity, the kinetics linking the exposure and the dynamics of the toxicity. And then and this will be given by Jos Bessems from Vito in Belgium. And then Anneli Julander from the Swedish Environmental Research Institute will talk about systemic dose of metals, the role of the skin, penetration as a reservoir, and the absorption of these metals. And then in a third talk, we will look into external, internal exposures of firefighters, instructors of firefighters, in fact, on polycyclic aromatic hydrocarbons. And this talk will be given by Jeroen Vanoirbeek from KU Leuven. So I now may introduce then the first speaker in a bit more detail. So why ADME and toxicokinetics are pivotal to known internal exposures, or as a missing link between exposure and toxicity. So this talk given by Jos Bessems from Vito in Belgium. He is a senior researcher at Vito. He is also a co-founder and a board member of ISIS Europe, as you can see, and is really involved in biomonitoring and biomarkers in general, and so is active as a supporting chemical management in EU since 1999. So I think he's well placed to give this talk, and as you can see, his domains of interest: tools for human biomonitoring, generating the use of ADME, which is, I think, very important in understanding toxicity and dosing, development of new risk assessment methodologies, and early warning systems. So I will stop presenting now, and I give the floor to Jos, please.
Can you see my screen now?
Yes, we can. Yes, yes.
Okay. Thank you very much, Peter. Thank you very much to the organizers for having me here. I really feel honored to be able to try at least to tell you a little bit about my past experience, which is the use of ADME and toxicokinetics knowledge in the chemicals risk assessment. I'm a toxicologist, a biologist by training. I did a PhD in chemistry in Amsterdam. After that, I've been working at TNO in the Netherlands on chemicals risk assessment. Then I switched to the RIVM, the Dutch Public Health Institute and Environment Institute. And in between what I'm doing now and RIVM, I spent 4 years in Italy, in Ispra, at the Joint Research Center of the European Commission, to work on the role of toxicokinetics in alternatives to animal testing. And since 2017, I'm working at the Flemish Institute for Technological Research, and my main interest, as Peter already mentioned, is the use of human biomonitoring in environment and health policy domain. So what is the overarching objective of EXIMIOUS? I had a little look because I'm not in EXIMIOUS myself. And I think it's a great project. Also, the EHAN network is, I think, very important. We are trying to contribute now also from the side of Vito to EHAN in general with research infrastructure. But the main objective is to assess the human exposome. So I thought that's really something interesting, because Paracelsus already said it's the dose that makes the poison. And my statement as a little change on that would be, it's the internal dose that really makes the poison. So what is the exposome? I'm not going to talk about that in detail, but I just wanted to show you this nice slide where you can see that it's not a very simple concept. You have the external exposome, you have the internal exposome, you have the endogenous exposome, the exogenous exposome, even transformation products of, in this case, chemicals are part of the exposome. And if you talk about endogenous chemicals like acetaldehyde or fatty acids, you usually talk about the metabolome, but that's a gray area, because in some papers you see that also metabolites of pollutants are taken within the metabolome. So you have to be very careful when discussing this to know what you're really talking about. And sometimes they might be the same. I mean, we know that ethanol is an endogenous substance. We produce it ourselves, but many of us also now and then take a bit of alcohol. So alcohol itself is an endogenous as well as an exogenous compound. And then we have, of course, also contribution of the gut metabolism to transformation of xenobiotics. This is just to give you a little bit of an idea on what are we talking about in terms of concentrations in the systemic circulation. So in the blood in humans, you can see that nucleotides, lipids, and steroids, that kind of substances, have the highest concentration when you talk about blood concentration. Where if you go lower in the figure, for example, the compounds that we are exposed to, but we don't produce them ourselves, it's the drugs, which is not strange, of course, because we really need to put them into our body in order to make sure that they reach a pharmacologically active concentration. They are more or less in the middle. Then substances that are in food, like food additives, food colorants, they are a little bit lower. And then chemicals that we see usually in cosmetics are even lower, and the pollutants are even in the area of nanograms per milliliter. Which, of course, and that is something that we have to really be aware of, the exposure itself is just one part of risk assessment, because some pollutants might be much more potent in terms of toxicity than some drugs. So even when you have them in lower concentrations, it doesn't mean that they are of lower risk. So the central question of this symposium today, as Peter already mentioned, is, do chemicals penetrate human skin? And how does skin absorption contribute to human exposure? So for that, I'm going to tell you a little bit about theory. I've just seen with a large group of more than 55 participants. And I guess that some of you are not very familiar with ADME and ToxicoKinetics. ADME standing for absorption, distribution, metabolism, and excretion. So I'm going to show you just a few slides to elaborate a bit on the theory. So, of course, we can ask the question still, why is it interesting? Because my statement is the internal dose that makes the poison. And often the concentrate, well, not often, actually always, the response is dependent on the dose. And usually you see a monotonic dose response curve, where if you increase the dose, the concentration in the systemic circulation goes up, and then also often the effect goes up. But that's not always. It might flatten in the end, where more dose or more exposure doesn't increase the concentration, and so also not the effect. It might even go, let's say, up and down. We all know, or we have heard about hormetic responses, where you have non-monotonic dose response curves, but I'm not going into that detail today. It's just important that you are aware that that might be relevant, maybe not so much for skin directly, but it's certainly something that you have to take into account if you are going into the experimental domain, where you increase the dermal dose with animals, where and I haven't because I had to limit myself with the number of slides, but just to tell you that if you increase the surface area dose, so the actual concentration per square centimeters, that at a certain point the absorption is saturated, meaning that if you increase the surface area dose, there's not going to be more chemical in the blood because it's saturated. So be careful about that. So this is probably a slide that you are much more familiar with than I am, because it's about acute allergic skin reactions. And this one is from skin sensitizers, where you really do see already in this figure that in order to bring your chemical in contact with antigen presenting cells, the chemical or a metabolite of the chemical, it might be a metabolite of the gut microflora, it does need to penetrate at least the first layer in order to get to these antigen presenting cells. Also here, this is an example from allergy. So this is an example of lung epithelium, where you also can see that penetration of the epithelium of the lung is necessary in order to start the first, let's say, event that in the end can result in allergic disease. Also for immune toxicity, adverse aggregate exposure pathways and adverse outcome pathways are relevant. This is a little step that I'm going to make now, but I think it's important because in the last 10-15 years, this has really been developed, and I think it's an extremely nice way of putting all kind of relevant information that we know in the toxicological arena, in the experimental animal toxicology domain, but also in the in vitro toxicology domain, as well as in the epidemiological domain, to put all these pieces of information together in a framework. And I'm sure that most of you have seen this, that when you go from a source to the target site, the source might be not one source, but many different sources. So we call that now the aggregate exposure pathway. And then when there is absorption and systemic circulation, so your blood brings it to the site where it can cause toxicity, the molecular initiating event might be triggered. And in between, that's what we call toxicokinetics. And for immunotoxicity, it might be much more simple. You're probably more familiar about that than I am. But for immunotoxicity, you don't need to go often much further down the road than absorption and then a distribution in the skin. So not through the skin, but in the skin to these immunologically relevant cells. So this is really what I want to talk about today. Really intermediate between aggregate exposure pathways and adverse outcome pathways. This is another way of showing this, that you can have many different sources of chemicals. The media through which these chemicals are brought to you might be very different, but in the end, they cause, or they may cause, if you're not protected, dermal exposure. And then absorption, the A of absorption, makes that the amount is going into the skin, and it might even go through the skin, and then the blood might take it to other organs in your body where so that's the distribution where these chemicals might be metabolized. And after all, you hope that they will be excreted as well. So that's the E of excretion. That might be via the exhalation. If you're talking about small molecules, it might be excretion via the feces. But even some chemicals are excreted via the skin. As you probably know that from some food constituents, you can smell it from the skin that you have been eating that. And altogether, we call that toxicokinetics. Toxicokinetics is about the concentration time profile of a chemical in the blood. So ADME is overall resulting in concentration time profile, and that's what we call toxicokinetics. I will come back to that later. This one I found very interesting to show you in terms of the aggregate exposure pathways, that if you don't know anything about the biokinetics, it's going to be very difficult to estimate concentrations, in this case of bisphenol A, a well-known chemical compound on which there has been a lot of discussion, not only in science, but also in the policy domain. So really very important to understand that the biokinetics is in between the aggregate exposure pathway and the adverse outcome pathways. And in this case, nicely presented here, bisphenol tissue concentrations can be indirectly measured by measuring the bisphenol A in urine or in blood, or even its metabolites. For example, the bisphenol A glucuronide, which can be measured in urine. And all together, you can say that this is the result of the exposure on the left and the toxicokinetics in the middle. Because if there would be no absorption, you would never measure bisphenol A concentrations in tissues and also not in urine. So absorption is the first step that is really needed in order to trigger any toxicodynamic reaction, even in the skin-related immunotoxicity. This is just another one to show you. Do you see this, by the way? When I'm
Yes, we see the picture and your mouse moving.
okay, so I see a black box, but I'm moving this to my laptop then. Okay, so it was a little bit disturbing, sorry. Just to show that this way of thinking, taking into account the toxicokinetics, is also essential for mixtures. These are aggregate exposure pathway networks for natural and synthetic dietary estrogens. So you can see here on the left, we are talking about bisphenol A, for example, that might be present in canned food, in handled food, in dust, and then you might be exposed via food, but also via air. And in the end, with the absorption and distribution, it leads to certain tissue concentrations where it might trigger the first event, the molecular initiating event for the AOP. The same is true for phthalates that are often present in plastics, but also in fragrances, where you might have exposure via the food and via the skin. And it's the ADME and the toxicokinetics that determine what phthalate tissue concentrations are going to occur. And the third one here is isoflavones that are present in the soybeans, for which the exposure is mostly via food. So what is ADME and what is toxicokinetics? When preparing this presentation, I went back to my own work and I found this nice report that I produced when I was working at the JRC, and I think it really nicely illustrates on the cover page what it's all about. So we are talking about exposure, and that can be in the field, in this case where an applicator is spraying some kind of pesticide. So you might even have skin exposure because you can see that this person doesn't have his hands covered. But for some other applications, your exposure might be via your lips. And then absorption, distribution, metabolism, and excretion all together result in a plasma concentration time curve. And that is depicted here nicely, where in the beginning you have mostly absorption, slowly also distribution starts, but at the same time, once the molecules are entering into the kidney, also excretion might start. At a certain moment, there is no absorption anymore because for example, your dose on your skin is gone. So then it goes down, and then at the end, only excretion is active. And in the meantime, you also might have some metabolism, obviously. You can see it here on the right a bit better, where it's the exposure, the anatomy, and the physiology of your body that determine together with all kind of mechanisms, active or passive, that you have in terms of absorption, distribution, metabolism, and excretion, what your final in vivo concentration time curve is. And in sake of time, I will just skip this one. This is just to show that you can measure many of these processes also in vitro. But if you want to predict in the end what is the concentration time curve in a human being or even in an animal, you need to put it into a model, what we call the physiologically based toxicokinetic models. But to feed into these models, you need information from different in vitro tools like in vitro skin permeation, or here microsomal or hepatocyte incubations to measure the metabolic parameters. And in the end, it will either give you a very low concentration here on the right, where it doesn't reach the level where toxicity occurs. It might give you toxicity when the amount is a bit higher, or when the amount is even much higher, it can lead to severe toxicity. And you can see here also directly on the lower one that the exposure scenario determines also your internal concentration scenario. So you might have a few exposures, for example, every day, then you get high peaks, but you also might have smaller exposures per occurrence, but with higher frequency. So several times during the day, for example, when a chemical is in your food and you eat 3 or 4 times a day, this will more or less be the pattern. This is a few terms that are important when we talk about ADME and toxicokinetics. So this again, this is again the line that describes the concentration. So on the y-axis, the concentration over time. This one going up and then going down when elimination, excretion takes over. And the area here, we call it the area under the curve. And the area under the curve is really a very important parameter to compare different exposure routes with each other. So this is called the maximum concentration that is reached, and this is the maximum, the time at which the maximum concentration is reached. So here again, a few examples of different exposure scenarios. This is every 8 hours. So for example, the meals, this one is once per 2 days, so once per 48 hours. And then what you can see nicely here, and that's also an important parameter, it's the half-life. The number of half-lives determines how much is remaining. So if you have gone one half-life, there is 50% remaining. If you have gone 2 half-lives, there's 25% remaining. And as a rule of thumb, usually it's assumed that if you have gone 5 half-lives, then the concentration is almost zero, because then it's more than 95% of your chemical has been eliminated. This is a picture to show you the difference between intravenous administration and oral administration, where obviously when you give intravenous administration, the concentration is directly very high. And when you give the same amount via gastrointestinal tract, the concentration is increasing much lower. And then in the end, of course, the excretion will be the same. And this one, but that's maybe already a little bit too much for now. If you're interested, you can always send me an email. This is to calculate the difference between the area under the curve when you do oral dosing and the area under the curve when you do intravenous dosing. In this case, you can see that the oral dosing gives you a much lower area under the curve than the intravenous dosing. And this is a term that is often used in pharmacology to really give the pharmacotechnological, let's say the pharmaceutical people, an idea about the efficacy of absorption of a molecule. So if the absorption is low, for example, only 10% of your chemical is absorbed, then the F value, the fractional bioavailability, can never be higher than 0.1. It's always expressed between 0 and 1. Then the next topic, immune response. It's not just a local topical reaction. some people think that immune response is just topical, but topical is without really penetration. For example, some chemicals can cause irritation or corrosion, and that's usually seen as occurring without really absorption through the skin layer. But the question then is, do chemicals penetrate the skin after all? Because if they don't penetrate the upper layer, then they will not even reach the cells that are the first cells important for the immunological response. And the answer is yes. Chemicals like mercury, isocyanates, PCBs, acrylates, and a lot of pharmaceuticals are penetrating the skin. And for pharmaceuticals, of course, they are designed to cross the skin because they are applied via creams, for example. And usually it's the low molecular weight chemicals that have good solubility in water and fat that penetrate the skin more readily than large, highly hydrophilic or highly lipophilic compounds. Although reduced integrity, etc., may very well influence the absorption. For example, the hydration level of your skin is important. The gender makes a difference, but also the health condition or the sweating rate can influence the amount, but also the rate of absorption of a specific chemical. Just another slide with a lot of chemicals and a group of chemicals that have what we call a skin notation. A skin notation means, that's a term that is used in occupational settings, that under the usual occupational settings, the amount of the chemical that is absorbed via the skin is higher than the amount of the same chemical that is absorbed via inhalation. For inhalation, there is often determined a concentration that the workers might be exposed to without health effects. But what has been observed in the last 40, 50 years is that for many chemicals, it's not so much the lung absorption that causes toxicity, but it's the skin absorption. So they have a skin notation, and it's a warning for the risk managers, the occupational risk managers, that for these chemicals, you really need to use personal protective equipment like gloves and sometimes even masks to protect your skin in your face against these chemicals. So this is again a nice slide to show a lot of factors that might influence the absorption. This is a nice slide to show that you have various routes of penetration into the skin and through the skin. It might be going between the cells. It might be going transcellular, which is through the cells. This is more the case for more lipophilic chemicals, because then they have to penetrate at least the cell membrane. And if you want to know more about this, all the links are available. So I think the presentations will be available on the EXIMIOUS website later on. At least my presentation is fully available. Then we did some work on modeling, because as I mentioned, a lot of work nowadays is done with in vitro assays, because it's more and more clear that animal models don't perform that well because the animal skin is different or the animal metabolism is different. So PBPK models are set up for not only oral administration, but also for dermal administration. And I'm not going into detail for oral administration, but even the gastrointestinal tract does contain or is built up by different parts. And in the dermal administration, you need to take into account the transport via the hair follicle. If it's a quite hydrophilic chemical, if it's more lipophilic, it's going through the stratum corneum. And all these parts you can more or less study in vitro, and then you will end up with some absorption rate constants or penetration constants, and you put them in your PBPK model, and then it's entering here. So then you can the model will give you the concentration time profile in the plasma, and that's what we did with a well-known compound, which is caffeine. Caffeine is more and more used in dermal cosmetics, like some shampoos. And what we modeled when I was at the JSC is just a single dose, and you can see here what we modeled as a concentration in blood. and what does happen if you apply multiple doses. You can see that the concentration goes up, and that also the half-life is such that after 1 day, there's still a considerable amount of caffeine in your blood if at time point 0 you applied it. So it takes more or less 2 days before everything is gone. So if you apply it several times during the day, it will take a long time before everything is gone. I'm not saying that it's dangerous, but just as an example. And then it's good to be aware that the absorption rates can be very different in different parts of your skin. For example, if you compare it to the forearm, where the absorption rate was set at 1, then you can see that in the palm of your hands, it's already a bit faster. But certainly your forehead, where your skin is very thin, and your scalp, the absorption rates are much higher. And that is probably why caffeine in shampoo really is going into your blood quite fast, faster than when you would have something with caffeine on your hands. And this is interesting, the scrotal area, because that was one of the well-known examples in toxicology where chimney sweepers in the UK appeared to have scrotum cancer in much larger incidences than other people. And after some research, they found out that the chimney sweepers sometimes scratch their scrotum. And because they had all these polycyclic aromatic hydrocarbons on their hands, the exposure of the scrotal area was very large, and these PAHs were absorbed very rapidly. This is just an example to show you how you can work with decision trees. I'm not going through this, and it's also set up for inhalation. But I think many questions are relevant also for dermal absorption. So if you have time, it's really interesting to just to have a look at this. Then the last one, and I think then I'm within my time. There are models available for in the public domain, and this is an example of a model at the US EPA, where you can model the skin exposure and what will be in the end the concentration time profile and your biologically effective dose in your organ. So with that, I'm happy to answer any questions. Thank you.
Thank you, Jos. Very interesting. So I have a look here in the Q&A. I do not see directly a question here, but just to give the audience a feel of the models, because you showed a lot of models, PBK models, and that you can have in vitro, so meaning cell cultures being exposures, linking that to internal doses in humans or in animals. So how good is this predictability of these models? Is that almost perfect, or is that what is the error range? Do you have an idea of that?
It can go from, let's say, directly within a factor of 10 if you have also the availability of real life human data, but you can also err several orders of magnitude. That is something that you have to check, but you have to start somewhere. And the nice advantage of using PBK models is also that you can turn the knobs. You can squeeze. So, for example, you can just in your model assume that the absorption rate is higher, and then your model prediction will tell you what does it cause for your concentration time profile. And then you will see that for some chemicals it doesn't differ that much if you increase the absorption rate with a factor of 2 or 5, that the concentration time profile will not go up significantly. And that might be because your elimination is much faster. So every, let's say, molecule that is entering the liver is immediately metabolized to a metabolite that can be excreted. but it can also be the other way around. So you always need to validate this against some in vivo data. And what is sometimes done is that you do in vitro animal testing and then an in vivo animal experiment also, and then this similar human in vitro assays, and then you try to, let's say, make a parallelogram. If your animal PBK model predicts well, then you have in general more certainty that your human PBK model with its similar setup will perform also better than if you don't know anything.
Yeah, no, I understand. So it's really a kind of ballpark that you're in, that you can see, and as a first step in the relation absorption, excretion, and so on. That is correct, but the exact altitude of the internal exposure, that is still to be checked, if I understand right. But I've got also a question here on the chat. So do you have, no, you have shown an example of exposure risk analysis method. What is the advantage of this method over others? So that's a question of Munir Jama.
So what I understand is the question: what is the advantage of using specifically PBK models?
It's an to be honest, exposure risk analysis method. That is what you have shown an example of that.
Okay.
Oh yeah,
Yeah.
yes, is yeah. So
well, the
you're right.
advantage is that you can simulate real life, and that can learn you a lot. Sometimes you have access to data. For example, in occupational settings, often measurements are being done for concentrations in air, and also human biomonitoring is done. So you can use the air monitoring data to feed your model. So what has been the exposure of your worker, and then you can use for that worker also what do we know, what the human biomonitoring data. So first you predict what you will see in urine or sometimes in blood, and then you compare it to what you know about this worker. And I really would have liked to show you some examples, but 20 minutes, it's really too short to do that. But feel free to send me an email and I can send you some examples.
Okay, very well. Thank you. I do not see anything in this chat and in the Q&A at the moment. So,
there is one question from
yeah, there is
Cam.
one extra question. So, can you discuss how babies and children's skin absorption may be different from adults? Is there much research on that?
Actually, I don't know, but I would say that in general, what I do know is that, for example, that's research that has been done with neonates that are still wearing diapers, that under the diaper, the skin is sometimes irritated and might be much more, let's say, penetratable for several chemicals. I know that I think it was in France that they have also put a ban on the use of some chemicals in diapers, because it was shown, if I remember correctly, that some of these chemicals were really penetrating the skin, and that they measured them already in the blood of these babies.
Okay, thank you. So yeah, and as you said, it is very brief, this seminar, and so I see that is in fact also time for the next slot. So thank you, Jos, very well and very clearly explained the ADMI and toxicokinetics. Thank you. So
it was a pleasure. Thank you.
Then I come to the next speaker, and that is Anneli Julander from the Swedish Environmental Research Institute. So this senior researcher, as you can read, and in work and a work environment expert, has a PhD in environmental sciences from Örebro University, and is a teaching occupational environmental medicine at the Karolinska Institute. And so her interest domain is interaction of toxic and allergenic chemicals with human skin and its local and systemic effects. So that is really fitting very well in our talk for today. So please, Anneli, take the slides you can present. I stopped sharing, and please, you may commence your talk.
Yes, good afternoon, everyone. I hope that you see my slides now correctly. And
yes.
To give some insight, a quick thing about the baby skin, actually from the age of half a month to 2 months, the skin barrier functions as the adult's skin barrier function. So just a quick note on that. So my topic of today is the systemic dose of metals, the role of skin penetration, reservoir and absorption. And I would also like to thank the organizers for giving me a chance to talk on this specific subject. I'm a docent, which means I are certified to teach, but it's also the equivalence of an associate professor in other countries. So skin, why should we care? Well, Jos gave us a good introduction to it, but let me tell you a bit more about this rather interesting organ. So it is actually an EU slogan that said the most important 2 square meters of your life, because it is the ultimate defense against our outer environment. We also have a barrier against water loss, and this barrier is really important when it comes to the penetration of chemicals, so we will get back to that. It also, for example, synthesizes vitamin D. It affects the immune system, and it's also coupled to our mental health. It's an indicator of health and emotions, and it is renewed every 4 to 8 weeks. So it's very much an organ that we can see a lot of things from. So if we move into the epidermis and the barrier function, because this is what Jos also said, the chemicals has to penetrate the skin to actually come in contact with our immune system and will furthermore also absorb to the blood. So in this situation, it is the epidermis that stands for the barrier function of the skin. And the epidermis can have different thickness depending on where we are on the body, as Jos also showed us. And it does regulate the transepidermal water loss, the TUL. Here is the picture of how the epidermis works. So inside our body, we have approximately 70% of water. And in the outer layer of the stratum corneum, we're down to 15%. So what happens with the stratum corneum? It's a very thin layer of dead skin cells. So it's approximately 10 to 20 micrometers thick. And they are dead, these cells, so no nucleus is present in it. They don't really have a cell membrane. But what is cool about the stratum corneum is that it can absorb 3 times their own weight in water, which means that when you soak in your bathtub, you get all wrinkly on your skin, and that's why the stratum corneum has taken up a lot of water. But if the stratum corneum would contain less than 10% of water, it will actually degrade and it will lose its barrier function. So it will open up for more penetration of chemicals. So that's why it's so important that the stratum corneum is functioning correctly. So this was a brief introduction to the barrier and the skin. So now let's go over to give the same brief introduction about metals. Metals, as you probably know, are ubiquitous, and we come in contact with them several times every day, our entire life. And when we start thinking about where we can come in contact with metals, these pictures are quite common. So, from training in the gym, having our cutlery, scissors, coins, mobile phones, computers, doorknobs, if we play instruments, keys. So the metals are actually really all around us all the time. So then that makes us think metal skin contact. I would say that skin contact with metals often comes from the materials of the items that we touch. And these materials are often pure metals. They can be alloys, which means that we have a mix of metals. in the material, and they occur on surface coatings. It can also be from dust particles and friction, like machining or welding. But then metals are also produced as metal particles, and we will get back to that with an example of what happens to skin with metal particles. Skin can also come in contact with metal ions when these have been released from the items. Metal ions can also be present in different types of oils, solvents, and cutting fluids in the working environment. And the problem with metals is, I'm sure you know, that they are carcinogenic, allergenic, and toxic. And in this presentation, I will focus on 3 metals. So it's nickel, cobalt, and lead. But how does it work? And luckily, since we have a time limitation, Jos showed you this picture. So this is slightly the same picture on the different absorption routes that a chemical can take. But for it to be absorbed, first we need the exposure. And the exposure to skin is dependent, as for many other things, the frequency, how often it is exposed, and the duration, for how long. When it comes to metal, I think you've all heard of nickel allergy, and probably affecting the ears if you have earrings, and that's a very long-term duration. But we also have all these short-term duration exposures when we touch our phone or our computer or our keys, etc. But it also depends on the skin status, and specifically the epidermal barrier, as I explained earlier, and also on the chemical properties. So when it comes to metal, how easily does the metal release ions onto your skin? So skin absorption is basically a process of penetration into skin, and after that it has to do with the permeation, and that's just diffusion processes basically. But what is often forgotten is that we may have a reservoir effect in the skin, and that is what is left in the skin. And often this is a concentration that will increase over time, and it causes different types of effects in the skin. For example, it can sensitize you so that you become allergic and have eczema, or it can have toxic effects. But if we then move on to how we can measure this exposure and the absorption. So the first slide that I want to show you is the in vitro studies for skin absorption, and it's actually an OECD test guideline that you can follow, and it's based on diffusion cells. These are known as Franz cells. They consist of 3 compartments. So we have a donor compartment up here that is illustrated in these pictures like little particles floating around in a solvent. We have the skin compartment, which is the orange layer, and then we have a receptor compartment which should mimic the blood. So it's usually a PBS solution in here. When you run your experiment, you can run it for up to approximately 24 hours. Some experiments can go all the way up to 48 hours, but then the skin is really not functioning well any longer. And it's recommended to take 6 to 12 samples during 24 hours exposure periods. And to take a sample, you actually have to have both, you sample through this sample arm here. So you have to both take out receptor fluid and put new receptor fluid in at the same time. So it does take a bit of handling in the lab. But it's a very nice setup to measure penetration of chemicals. And from this type of studies, we know quite a lot about different organic chemicals, but they haven't been used so much for metals. I will give you some examples of research that we have done further on with these diffusion cells. But if we don't want to do the in vitro situation, we can actually do quantitative skin sampling in vivo. And it's not that difficult, at least not for metals. So if we want to measure on the skin surface and to get a skin surface dose, a very common method to use is wipe sampling, which is basically we wipe the skin with a dilute acid or water. Or we can also use a method called tape stripping, where we do, it's quite simple. You put a tape onto your skin like this, and then you remove it. And then you put the next piece of tape on the same surface and you remove it, and you keep going. So if we tape strip 1 to 3 times, we will get an answer of what is on the surface of the skin. But if you then tape strip 3 times and up to 20 to 30 tapes from the skin, then we can start saying that we are measuring in the skin, and then we get to the skin concentration or the reservoir in the in vitro studies. But if you don't want to follow a chemical tape by tape so that you can get a nice graph, you can just do a skin punch biopsy, and then you get the entire skin out. So that is quite an invasive technique, actually. So it's mostly performed by dermatologists looking for other aspects than chemicals in the skin. But if we then move into this penetration, reservoir, and absorption situation, I thought the easiest way of explaining it to you is to give you examples from scientific studies with inorganic metals. So these, I will give you examples of first nickel, then a couple of examples for cobalt, and I finish off with one for lead. And at the end of this, I hope I've shown you that absorption of metals do occur. So in the first study that I would like to talk about is nickel and the skin, and it's a penetration study, and we published it in, well, actually last year. The background for this study is that we know that when it comes to nickel, it only takes 3 seconds of a contact with an item for us to have nickel metal ions on our skin. If we look at dermatitis patients, we have reports of them telling us that, you know, if they put earrings on, for example, that contains nickel, they will react within 10 minutes. So it's quite a quick reaction, and this hasn't really been known before, because contact allergy is considered a late allergy, so it takes a few days for the allergy to get started. But some patients do report symptoms already within 10 minutes. And we have a gene, it's called the filaggrin gene. I don't know how much you know about it, but this gene does regulate how well our stratum corneum and epidermis has its water content. So if you have mutations in the filaggrin gene, you will have some difficulties with your water retention of your skin. So you will have an impairment in the barrier. And the same goes for people who suffers from atopic dermatitis. So we wanted to find out how the filaggrin gene affects the penetration of nickel. So we did a study with this purpose. So first we had to screen some 445 persons for the filaggrin status, since the prevalence in the Nordic countries is about 6, 7 to 10% with filaggrin mutations. And in the end, we could include 40 persons in this exposure study. And we divided them according to the filaggrin status. So we had 17 persons with the filaggrin mutations and 23 without. We also asked them by using the Nordic Occupational Skin Questionnaire that is developed in 2002 about their skin status to see if we could screen out people with atopic dermatitis. After this, we applied nickel on their lower arm. And after 4 hours, we started to remove where we applied the nickel by tape stripping with 10 consecutive tapes. So the result of this study is shown in these graphs. So here we can see nickel in microgram, and these are the tapes, tape 1 through to tape 10. The black ones are filaggrin null, those with mutation, and the more gray colored bars are the wild type, those without mutation. So what we quite clearly can see is that for the gray bars, we can recover more nickel from the skin than we can by the black bars. So this means that the nickel that we applied in 4 hours, it has penetrated below the depth of what we can recover with the 10 tapes. And if we then looked at their skin symptoms, people with skin symptoms are then the black bars, and gray bars are no skin symptoms. We do not see the same difference in the tapes of recovering nickel. So at least for the penetration of nickel, we can say that this rather small study, though, indicates that if you have a filaggrin mutation, nickel will penetrate much faster into your skin. So the next study that I would like to talk about is cobalt and skin absorption. And the background for this study is that in hard metal facilities, cobalt is used as a raw material. And cobalt have, if you look into the literature, you will see a lot of papers, mainly from the 90s, that cobalt is suspected to be absorbed by skin. And as Jos said, we can often go out and measure air, urine, or blood, and this is what most of the studies have done. So no one had really focused on measuring skin exposure. So what we did was that we got in contact with the hard metal industry in Sweden, and they did have somewhat of a problem because they told us that we have workers that have elevated urinary levels of cobalt, they have no air exposure. So this is what we wanted to understand. Could the skin be the missing link in this exposure? So we included 76 workers with exposures, and they were grouped into raw material or finished product workers. And the raw material in this case is either powder or pressed shapes. And then when you produce this, you send these press shapes into big ovens and in something called a sintering process. And then we have these beautiful finished products that can be used. And this material is very hard, so it can be used to drill into mountains and for cutting tools to cut other type of metals with. We did a collection of skin wipes, personal air samples, and urine sampling. And what we saw was that, as we thought, we have higher urine concentration and skin concentration in the raw material processes. And that's not surprising because the exposure is much higher. We have both skin exposure and air exposure. But what was also present was a clear association between our skin levels and the urine levels in the workers that work with the finished products where no air exposure occurred. So all the air samples collected from finished products were below LOD when we analyzed for cobalt. So there were actually no cobalt in the air there. This kind of shows that we have a strong association between the skin and urine levels, indicating skin absorption. But I wouldn't say that we proved it, but it looks extremely likely that cobalt do penetrate the skin all the way, so that can be traced in both blood and urine, actually. But these workers do have a lot of powder exposure, we thought it would be even more interesting to also look at how this cobalt powder might affect the contact allergy risk. So we set up a study where we are investigating cobalt nanoparticles and contact allergy. And in this study, we want to see the difference between particles of cobalt versus ions of cobalt. A little bit of the background for this study is that we have it's quite common to be allergic to cobalt. So in the general population, we have up to 2 to 4% that has a contact allergy to cobalt. And as I said, cobalt in particle form occurs in hard metal production, but also the growing additive manufacturing or 3D printing uses cobalt in nanoparticle form. So it's of importance to understand how this works with the skin. And here, this is a really small study because when it comes to testing people with known allergenic properties, we cannot use huge numbers of people. So we included 14 persons that already had a cobalt allergy and tested these people by patch testing with cobalt nanoparticles and cobalt chloride. And for those of you who don't know what patch testing is, it means that we put a little bit of the substance on a patch that is taped on the back of a person. So it's not like a prick test of allergy where you actually prick your skin. So this is only a skin test with an intact barrier. Then the result is read at day 2, day 4, and day 7 to evaluate if we have an allergic response. As you can see in this picture here, we have a nice eczema picture of cobalt nanoparticles. And then we also collected biopsies at day 4 so that we can look into staining and histopathology and realize that, as we can see here, we have a lot of infiltration of cells into the skin. So it is a true allergic reaction. So the result that we have from this study is that the nanoparticles will give rise to eczema, and as measured by the positive patch test. We also saw identical responses for the cobalt nanoparticles and the cobalt chloride in a set of immunological markers that we measured. And what was interesting is that the dose on the skin surface underneath this patch test, even though we tried to have the same dose, we did not achieve it for the nanoparticles. So we had much lower doses of nanoparticles on the skin, and it still elicited to the same level as the cobalt chloride. And we also collected biopsies to confirm by chemical analysis that we actually did have cobalt in the skin. So this is a very important study, and we think it's also important to avoid exposure to cobalt nanoparticles in these new occupational settings, especially within 3D printing. Then I would like to show you the last study that we have done, and that is on lead and its occupational exposure and an in vitro study. So here we started combining all our methods that we have to study it. And the background here is obviously that lead is a highly toxic metal. It's also strictly regulated in occupational settings, both with limit values for inhalation exposure and also in blood lead levels. So we had a case study that workers had very high blood levels, although again, no air exposure above the occupational exposure limit. So just the same setup as for the cobalt in the hard metal facility. So even here, we collected skin wipe samples, blood samples. We measured the air that the workers were breathing. And it's a case study because we only have 7 workers here, and they are CNC workers, so they work with cutting fluids. We also collected samples from used cutting fluids and analyzed the lead content and used them in vitro testing. So what did we find? If we start with the results of the occupational exposure, exactly as the company said, all the air samples were below the occupational exposure limits, both for the amount of dust in the air, but also for the lead content. So it was, if I remember correctly, less than 10% of the occupational exposure limit value. So it's quite low levels in the air. But what we clearly could see is that we have a lot of lead on the skin. So this axis has lead in skin in microgram per square centimeters. This axis is the blood concentration in microgram per deciliter. And here is the left hand and the right hand. And we can then see also that there are a large difference between different skin surfaces on our hands. And even though it's only 7 people, we just wanted to plot and see if there seemed to be an association between the skin and blood levels. And as you can see, it is somewhat of an association. And if we then go to our in vitro results, remembering this front cell diffusion cell that we had, where we dose, we have the skin, which is the reservoir, and then the receptor. So if we only look at what happens with the skin during the time points when we run the experiments, we see that the skin is accumulating lead over time. And but in rather low doses, actually. So if we were calculating how much of the applied dose that was retained in the skin, it's about 2 to 4% after 2 and 4 hours, and then up to 10% after 24 hours. And there is actually no collective knowledge about what happens to this reservoir. There is one study by Professor Larias Filon in Italy that shows that if we wash off skin within 30 minutes after exposure, it actually did not lower the lead content absorption over 24 hours. So something happens with this reservoir, and we need much more studies on this. But if we then also quickly look at this picture, this is then the receptor, which should mimic the blood. And what we can see is that we have extremely low concentrations of lead in this receptor. Again, this is microgram per square centimeter. Remember, the skin was approximately 20. And here, you know, the highest one we have is 0.043 maybe. So of the total dose, a very little amount of lead does absorb through the entire skin. But is this a dose that we don't have to care about because it's so small? Well, I would disagree. So we actually did a little bit of calculation. So we based our calculations on the US EPA adult lead model for soil, because that's the only one that we could mimic to skin exposure. And we fed the model with a lot of data then on worker skin concentrations, the air concentrations. We took the absorption rates from the in vitro study, and then we looked in the literature to look at uptake of lead from hand-to-mouth behavior, as well as how much lead is taken up via gastrointestinal uptake, and we used the skin surface area of 2 hands. So when we started doing the calculation, we noticed that if we have a hand to mouth going like this, if we have lead on our fingers and we get it into our mouth, we will increase the blood lead level with approximately 16 microliters per deciliter. The skin absorption from the in vitro cells says somewhere 3 to 6 microgram per deciliter, whereas the inhalation in this case only contributed with 2 microgram per deciliter. But please remember that all these inhalation concentrations were below the OEL, so they are supposedly to be safe, but maybe not so safe. And to give you some set of reference numbers for this, we can mention that the biological limit value for blood lead in Sweden are these numbers, and here they discriminate between women of childbearing age. So women that are under 50 years, they can only have 10 microgram per deciliter in the blood. And men and women above 50, the limit value is 30. So I think this is quite concerning, given that the hand-to-mouth behavior would be 16, skin absorption in these numbers. So I would say that we are not in a comfortable zone and a safe zone as concerning lead and exposure in this brass foundry. So to summarize, I think that we, through our research project, have shown that there is a skin barrier effect, especially if you have a filaggrin mutation that may affect the uptake of nickel. And we have also done studies for organic chemicals that shows the same. If you're a carrier of filaggrin mutations, you have an impaired skin barrier. Skin disease such as atopic dermatitis or contact allergy may affect the penetration, but it's not so clear as for the filaggrin. We do show that skin absorption of inorganic metals do occur, although it's a slow diffusion process and we have low percentages that are applied, but given the large surface area of our skin, it may contribute substantially to blood and urine levels. And we also have a clear reservoir effect. However, it is unknown how this contributes to absorption, so we need more research there. And the problem with the reservoir is that it's making metals available for the immune system, and especially these metals that are known skin sensitizer. This is extremely problematic if we walk around with nickel, chromium, or cobalt in our skin for a prolonged period of time. That was all that I was going to say, so I would like to say thank you for your attention. And if you have any questions that I can't answer here, you can also email me. Thank you.
Okay, thank you very much. Very interesting. I have a look here in my, in our Q&A. Do not see a question yet, but I had a small remark. Maybe the lead problem, if I look back to the presentation of Jos, it is, if I am right, I think lead has a relatively high or long lifetime, isn't it? So it stays long in our blood. So even if the exposure is low, but if you're exposed every day, it really accumulates, I think.
Mm-hmm. Yeah, it does. It does accumulate over time. So that's the problem if we start factoring also in skin that we haven't thought of before.
Yeah. Okay, so I see that there is something in the chat. Here, you said you found cobalt nanoparticles within the skin after batch testing. Which method did you use to detect it, and how far down in the layers?
A bit unclear. What we found in our biopsies so far that we have looked is we put them through the ICP-MS, so we just know that we have cobalt in the skin and not the nanoparticles. However, we are now looking into some fancy microscopy techniques to see if we can locate also the particles.
Yeah.
But I would think that they are in the very upper part of the stratum corneum epidermis. There are no indications that they would penetrate further down.
And how small were these particles?
In the range of 4 to 100 nanometers.
Okay, yeah. Okay, thank you. So it was very interesting, and yeah, I'm learning a lot today, so that's very nice. And I see that we are just in time, so and I don't see any. Oh, there was an extra question, so sorry. So from Jos, if I'm right, it's a bit. So thank you. So would you say that many metals stay long in the skin, and because of that, there is a lot of time for molecular initiating events to occur and so on, in, for instance, sensitization?
I would say that we hypothesize that since metals primarily in ionic form are good at binding to proteins. So we think that is what's going on, but we do need more research. But we are suspecting that they have quite long retention time.
Yeah. And a final question on temperature. So the France model, I will say, is carried out at 32, 37 degrees, if that's correct. So, and do you believe that temperature plays a role here?
Yes, it definitely does. So we carry out our below 32 because we have colleagues that shown it that if we carry it out at 37, especially for the longer time points, like 24 hours, the skin will start to disintegrate on top of the stratum corneum. So it does do a lot of damage.
Yeah.
So you have to be very careful keeping the temperature at the right span.
Okay. Very well. Thank you very much. So, and then I go over to the next speaker and try to find that slide. Yes. So the next speaker is Jeroen Vanoirbeek. He is associate professor and working at the Laboratory of Occupational and Environmental Hygiene here at the KU Leuven, so the same institute as me. And he has a PhD in biomedical sciences. So his interest is mainly in respiratory and skin exposure assessment, clinical epidemiology, and mechanisms in skin-lung interactions of exposures. So I will stop sharing, and so Jeroen, you can take over, and for you the floor. So I had here one or two things in the chat. First, an early question in the beginning of your talk about the measurement of test temperature for gloves at 23 degrees and an equivalent ASTM standard test at 35 degrees. So is there any work on the way to deal with this? So these differences to which temperatures to be used to do these type of measurements?
I know that the collective people that are doing in vitro studies, we recently published a paper where we comment on the OECD guideline, EFSA guidelines, and other guidelines, and saying we need harmonization about the temperature.
So can you identify yourself? Because I don't have a clue who is talking here.
This is Anneli talking.
Yeah, I thought, but I was not sure. Yeah.
Sorry. So I think there is work ongoing and trying to find out what happens, but I'm not aware how far it's come yet. Just that we have a lot of people recommending different temperatures. I don't know if Jeroen or Jos knows anything more about it.
No, I got the question often, and it's two sides to the question. One, if temperature increases, then humidity increases and uptake increases, and you get occlusion. So it needs to be taken into account. But also, if temperature and humidity increases, then the uptake of the chemicals on the measurement devices, like the bracelet and like the activated charcoal, are difficult. They are less controlled and they can be biased by that. So it's hurdles that we need to take to assess thermal concentrations of a chemical on the skin. And that's what I meant with one of my first slides, that it's not harmonized how to measure on the skin. And it's very difficult, and especially in occupational settings with warm or cold or humid, all kinds of environments.
Okay, thank you.
Can I contribute maybe, Peter?
Yeah, you may.
Yeah, what we have seen in work that we have done for the OECD, where we have set up a guidance for establishing human biomonitoring guidance values, is that in many cases we had some occupational hygienists in the group. It seems that human biomonitoring, where some people think it's really complex and expensive, it's less expensive than the other external monitoring. And the advantage is that you directly measure. You don't need to know whether it's dermal or inhalation. Of course, when you want to take risk management measures, you want to mitigate the exposure. Of course, then you need to know what is the major contribution, which route. But in general, I think the best way, and it will avoid all kind of complexities with respect to temperature and humidity, is the best to try to do human biomonitoring. And once you see then problematic concentrations of, then you can go back and try to find out more, I think. Thank you.
Okay, I think that Jeroen wants to answer to this one.
Just
Or
very short. You're completely right, Jos. But on the other side, if you measure the air, you can have 20 to 50 to 100 components in one measurement. And when you do biomonitoring, you have each analysis, you have a method to optimize, and you need, it takes a lot more time. So there are benefits and
pros and cons.
downsides.
Yeah, I agree.
I quickly step to a question here. So, Christer, did you do any measurement inside firefighter SBA masks during the firefighting, such as a wristband inside these masks? Did you try to do that?
No, that's something that we absolutely want to do, but I don't know yet how. So you could think of taking wipes on the insides of the mask. The thing is, with the activated charcoal patch that I saw, you could stick it underneath the mask. But with the silicone wristband, we don't have an alternative yet that we can stick, like say on the nose or something, or in this area. So it's something that I would like to do, and suggestions are welcome. But practical, I do not yet know how I should get that done.
Okay. And there was no question of you, Jos, but I see another one. First to that, thank you, Jeroen. Work presentation can penetrate the skin faster than usual considered. Allow me to post here a recalculation we published before. So it's just extra information. And you had a question, Jos, still on hydroxynaphthalene. Do you expect it could be used for other workers such as asphalting roads and so on?
If it's outside and it's a gas phase, then I don't know. I think hydroxypyrene is still okay, yeah? But not in the case of this type of exposure. 2 times 20 minutes with very high gas, with good protection against particles, and then less good protection of the skin against the gas phase. So it's a special situation, and I think that we need to look broader than only one hydroxypyrene, and depending on the risk assessment, what we know what comes into the air, then select the marker.
Okay.
Yep. Thanks.
Okay, good. Thanks. I don't see any other things coming up in the chat, so I quickly jump back to my slides. And I really thank you all three, Jos, Anneli, and Jeroen for this very interesting seminar. As I said, I learned a lot of things, and that makes it very interesting. Better insights, and the skin is certainly something that we have to consider in a better way. And I also thank the audience, a lot of people here today, so that's very nice, and hopefully we try to do 2 seminars a year, so hopefully we can see you again next time, trying to do that still before the summer, our next seminar, but you will find messages on that in time before to subscribe. And then certainly our local people helping me, Hilde and Manosic, for the support, but also people really at the back, communication partners helping us setting up the links, Emily and YY, and then certainly all our EXIMIOUS partners. We are doing very interesting research with together all 15 partners here, and again you see a map of Europe where we are located. So I thank you all, and I hope you had, as me, a good afternoon in this seminar. So see you, and bye-bye.
Bye-bye. Thank you.
Bye-bye. Thank you.